Thanks, David. So I thought I was going after Paige, so this is a good way not to be nervous before your talk. All of a sudden you're speaking. So I just want to, I know, and I had forgotten that Dan was going to actually cover some stuff, substantive stuff, so I'll skip a couple slides. But I do want to give this one overview. One more apology. I was cobbling together stuff on a loaner computer that did not have LaTeX or even Adobe, so it's like a ugly PowerPoint like this. So I want to highlight three aspects of genetic data that are, many people think of genetic data as one subtype in the big data revolution that's happening in computational social science right now. But I want to argue that they're actually very different and distinct from all the other forms of computational social science going on. So meaning, creating an API to download Twitter feeds, or scraping online message boards, or analyzing social networks about retweets or contagion on Facebook, and analyzing Google searches to get at what people's true preferences are. All of that's really interesting, but what's different about that is that I think that's very useful for revealing hidden information, particularly with areas of people's views that might suffer from social desirability bias. They say they vote for Trump for this reason, but actually when we look at search data, it reveals something else. But almost universally, that kind of data is looking at the electronic data as the tweets or the clicks or the likes or whatever as a dependent variable, as an outcome that's going to be studied. Even other biomarker data that you might have heard of, like epigenetic data, methylation of the genome, C-reactive protein levels as a measure of stress exposure, something as simple as BMI or blood pressure as a biomarker. Those generally are either the outcome, they're meant to reveal something about our health or something like that is more accurate than just asking people, or it's a mediator somewhere in a pathway from, say, the social environment to an outcome like education or mortality. And genetic data are very different in that respect. The only data I can think of in this suite of new forms of data that social scientists are utilizing that form the independent variable. There's three aspects of them that I think make them very useful for social scientists. First is that, as you'll hear about in Eric Turkheimer's famous laws of behavioral genetics, basically any outcome you can think of, even what kind of car you drive, even who you vote for, the big five personality types, you can go down the list, is going to display some minor, at least minor, probably moderate degree of heritability. Meaning, as we heard from Dan's lecture, that if we describe the variation in the population, a significant proportion of that variation in the population in the outcome is going to be described by the variation in genotypes in the population. So therefore, it's predictive genotype. Second, as we also heard, they're fixed, except for somatic cells that become cancerous through mutations, for example. Your genotype, which is an irrelevant exception in most cases of what we're talking about, your genotype is fixed at conception, does not change, so we know the causal origin point. We know that David's coming to NYU does not affect my genotype. So that's real, as any social scientist knows, that's gold. And then, as we also heard, conditional on parents' genotypes, every person is like thousands of little randomized controlled trials wrapped up into one person. And that's the other aspect that makes it amazing. And I say thousands and not millions, as you'll hear about later, because chromosomes travel in chunks, and it becomes, you don't have actually 50 million unique experiments. You have, I think it was on the order of 70,000 experiments that are happening in the chunks of the chromosomes that are shuffled in the parents during meiosis and then passed on to the kid at conception. So those aspects, I mean, what more could we want? I could throw in a Japanese ginsu knife or something like that if you act now, but this was like a dream come true for a social scientist who's interested in causal inference about a range of human behaviors, health, and so forth. So the one obvious way to use this information is to use it as a control variable. If we know that much of social outcomes, economic outcomes, political outcomes, health, other behavior has an environmental component that is influenced by the social or physical environment, but it's also influenced by genetics. Let's construct the polygenic score. Let's control for it. We'll have a baseline model without the polygenic score. We'll have the model with the polygenic score, and then we can see how the things we're supposed to care about as social scientists, that is to say the social environment, the effect of schooling on partisanship in voting, let's say, changes when we control for the relevant genetics. And we know we measure both of those imprecisely. We can start, there's of course lots of tricks to deal with that measurement error, and then we can say, okay, the effect of education on partisanship in the United States is actually much smaller. The treatment effect of an additional year of schooling is actually much smaller, maybe it's larger than we thought before when we control for the fact that genetics influence both partisanship and education level, if that makes sense. And I'm sure, as Dan will talk about, even if we have a randomized control trial, by soaking up a lot of the error variance that using the predictive aspect of the polygenic score, we can get more precise estimates of our randomized treatment. I won't go into all those. I want to talk about other ways to use the polygenic scores or genetic data in general that I think are very useful to social scientists. And I should say, I think all of these examples came from jointly with students who participated in this workshop. What's going on here? Not working. Dan, you know what's going on? There was a couple of alerts there I closed, but I don't. Okay, that works. Okay, these are the ones I'm now going to skip because we already know. Okay, so one first example I'm going to give is estimating peer effects, which, at least for sociologists, has been a bedeviling prospect. If we don't think that peers influence our behavior, we might as well shut down the whole field. And yet, that's an incredibly hard thing to show for a variety of reasons. I also want to say that, to back up for a second, I meant to say before that sociology is typically the field in the social sciences that is the most hostile to the inclusion of genetic data. I mean, the entire field was like founded practically against Galton, who was one of the trivia answers last night, the cousin of Charles Darwin, who started the eugenics movement, and so sociologists in particular are very skeptical and think that when you invoke genetic data, you're making a genetic deterministic argument, which we're not, or you're naturalizing and therefore legitimizing inequalities in society that are, in their view, almost entirely based on unequal social environments. So I should say that I do think that at least among more kind of normal science-oriented departments, that's changing, and social science, sociology departments are seeing that this is an incredibly exciting new form of data and are hiring people who do this kind of work and so forth. But there's still a lot of resistance, and I think using the genetic data in a way that I'm going to talk about, which is in the service of showing environmental effects, helps kind of calm some of that resistance down, and really what we have to add to the field. We're not going to, we're not interested in getting another, couple percent of R squared in a phenotype by better imputation methods of the genome or so forth. We're not going to. There are population geneticists and statistical geneticists that are working on things that have a comparative advantage in sort of identifying the genetic effect. What I'm going to argue is that we take that genetic effect and then apply it to social science problems, and that's where we can step in and have some value added to the entire field. So back to estimating peer effects. We have three forms of bias in peer effects. We have homophily. So if David and I both smoke cigarettes, did David influence me? Did I influence David? Or did we become friends because we were ducking out of that boring lecture and found each other in the boys' bathroom, and then we shared our cigarettes? The contextual effects. Did we both happen to start smoking not because again of mutual influence, but because of the fact that we both had this really stressful math teacher and we needed a nicotine fix. Or lastly, as Charles Mansky pointed out, the reflection problem. If we can rule out those other two, how do we know that it's in real time whether it was David that influenced me smoking or I influenced David? Even if literally I'm the first person who lit up a cigarette, maybe he dared me, who knows? We don't have the kind of, we're not inside people's brains, we don't have even the level of granular behavioral tracking that we would know the answer to that. So I'm going to argue that the genetic data helps solve these problems in combination with some other methods. And this is using the National Longitudinal Study of Adolescent to Adult Health that you guys are going to be using this workshop. And what we did basically, we have the question. You can look at this outcome as well if you like. During the past 30 days, on the days you smoked, how many cigarettes did you smoke each day? We've also done the 1-0, do you smoke at all currently? And we use a smoking polygenic risk score. I should note that this, unlike other polygenic scores, this is a score that predicts well in different ancestral groups. It still predicts the best among those of exclusive European ancestry, but it has much less of a drop-off, and that's because of the distribution of effect sizes. We don't have to go into all this, but the point is I'm going to show results that include, I've done it for whites only, but I'm going to show results that include all races for this particular study. So just to show the problem, this is the problem of homophily. If you look at schoolmates, people who say they're friends in the AdHealth survey, genome-wide, they are tend to have some they're not randomly assigned. They're more alike. So if you look at panel B, the educational polygenic score, there's a 0.11 correlation between friends. So the kids that are doing well in school and the kids who are not doing well in school tend to cluster together. This is across all schools. If you use school fixed effects, it drops about to about half. So in other words, when we look at who is friends with each other, half of that correlation is due to sorting that's already occurred into which schools they go to, and half of it is sorting within schools. Interestingly, there's no sorting on height polygenic score, and the BMI polygenic score is entirely within school sorting. It's not, there's no sorting between schools. So that just shows that you can't just take friends and look at their correlation and say, well, if friends smoke, then they must have peer influence. They could be finding each other. So what we do is, instead of looking at the friends per se, we look at the grade mates. That is, let's imagine this is like the 8th grade of a school. We treat it as random what the genotypic distribution of all of you vis-a-vis me is in the 8th grade, conditional on this school. In other words, like versus the 9th grade or the 10th grade, because obviously I've already showed there's sorting between schools based on neighborhoods and parental SES and magnet schools and whatever. But within that school, and we show this empirically in the SI, that it's random, the distribution of the smoking polygenic score by grade. So we use the grade mates solution. And by using the polygenic scores rather than the actual behavior, we know that it's not me influencing all of you, it's you influencing me, because I can't change your polygenic score. What do we find? Our preferred is the reduced form, where we just look at the metagenomic effect of everybody around us, and there is a positive effect of the polygenic score of my grade mates on my likelihood of smoking. We did a placebo with height. I don't know if any of you are familiar with the controversy between Nicholas Christakis in his famous paper on obesity being socially contagious using Framingham data, and then my colleague or co-author Jason Fletcher came around and used the same data to show that acne, headaches, and height were socially contagious, just to illustrate the fact that he had uncovered homophily most likely and not actual social contagion. So we do as a control height, which we do not think should demonstrate peer effects, and in fact does not. To put it in context, I mean, the error bar is much bigger because this is much more imprecisely estimated. But if you take the low end of the error bar, the effect is about 1/3 my own or half my own polygenic score for smoking. And we know that adolescence is a particularly, well, we think adolescence is a time that's particularly susceptible to peer effects. And you can see it compared to, say, age or the effect of being male, that it's not a trivial effect. We're underpowered for this, just to repeat what Dan was emphasizing, that you really should be adequately powered, but I can't resist putting this slide in, even though it's just suggestive, and we would love to redo this with a bigger sample size. But with the effects, you can then understand something about social dynamics in schools in theory. So we find that, for example, the biggest effect, again, they're all overlapping, so these aren't really different, but you could imagine redoing this analysis with a much bigger sample size, and you see that white egos, meaning the white respondents, the biggest effect on them is for white peers compared to all peers. And we find that non-white respondents are less affected by their peers. So that's all to say, once you establish this methodology, you could go in and even if smoking is not your primary interest, you want to understand racial dynamics in a school, you can use genetic data to help you do that. So then we want to ask, is it the average in my grade, or is it another function of the distribution? Is it, for example, what I think Carol and Hawksby called bad apples or shining stars in a different paper? In other words, do a few people who have really high genetic propensities to smoke influence the whole grade, or do the goody two-shoes influence the whole grade and restrain smoking? It turns out, this is basically the only bar you want to see, that the bad apples really matter. And that's the most, having a few people with very low genetic propensities to smoke does not help you. Okay, example 2. So I should have said that was joint work with Romina Sotude, who's just finishing her PhD at Princeton now, came to this workshop. I don't remember which one, maybe the Santa Barbara one or the Rutgers one. This second one, she's going to Oxford for a postdoc. The second one is from Asta Breithold, also participated in this workshop, and this came out of discussions here. So another question is, how do people influence each other in their families? Do we parent differently when we get a different kid? There's a famous joke that says parents of one child are environmentalists, and the moment you have 2 children, you're a hereditarian, because you see that, holy crap, like, these kids are so different. What did I do? Nothing. There's nothing I changed about my home environment. And moreover, the old school, I mean, Paige can correct me, but the old school of thought that came from Uri Brofenbrenner's model of the ecology of development in the 1970s was that there was the child at the center, then the immediate caregivers, the larger family, the community, and so forth, as like concentric circles, and the child is this like passive recipient of social influence. But now we know that the child is an active evoker of their environment. And there's been a large literature that I've been a part of that tries to identify that causally, but until now we've only had one variable that we can look at causally within families about how a child's characteristics affect their parents or their siblings or whatever. What would that be, aside from genetics? Gender, yeah. So there's a large and growing literature on that I think really started with Abania Washington AER paper looking at the gender of the offspring of congresspeople and how they voted, and arguing that those with female offspring tended to vote more liberal because they had more empathy theoretically towards females, and so they would vote more like in line with Emily's List and a National Organization of Women. Even across the Republicans and Democrats had this small treatment effect of having a daughter. I won't go into that whole literature of what's the state of that right now, but the point is now, because conditioning on parents' genome, we have a random draw that's not just their gender, but their polygenic score for smoking, their polygenic score for education, their polygenic score for BMI. You can go down the list, and now we have so much more information. We can look at how children influence their parents, how siblings influence each other, and so forth. And moreover, a classic sociological question is, how do parents with different means, that is, different socioeconomic statuses, parent differently? So Becker and Tomes argued that parents ideally reinforce ability, and that parents with a budget constraint can't do that. So therefore, parents who have more income will actually generate greater differences among their offspring, and parents that have a stricter budget constraint will mute those differences between offspring. I think sociologists, including myself, have argued that actually parents with more resources invest in a different way. They invest in a compensatory way. So in other words, if you're really rich and you have 2 kids and one is struggling, you actually invest way more in that kid to bring, in terms of other resources, tutoring, et cetera, to bring them up to speed with the baseline of the family and the expectations of the family. So that actually, and those with a budget constraint don't do that, and in fact have given limited resources, bet more on the kid with the most promises. So they invest in a more efficient but inequality producing manner. So again, 20 years ago now, I guess, God, I was involved in this debate, but all I could look at is the outcomes. I could look at how much did kids from different families resemble each other in the end in their attainment. But now we can actually look at what the actual process. And how we do that, of course, is by conditioning on the parents and seeing how the random draw of their offspring affects their behavior. So let's say David and I could just keep using David as an example. We could reproduce with our two sperm. Somehow, I'm sure that's going to be possible pretty soon. And let's say we're both at the 50th percentile for education, polygenic score. So, we could get a 60th percentile kid. We could get a 40th percentile kid, right? So that's what we're identifying the effect of, the draw conditional on your, and so, sorry, Dan, my ears must be bigger than yours. And the first thing we do is to show that it's what we're not detecting something that is population stratification or any of the other things you've heard about earlier. We look at age 6 months, and we have 3 samples. I won't go into the details, but the presumption is that at age 6 months, a baby isn't expected to talk yet, is barely, just maybe rolled over. There's not a lot of evidence to the parents that this is, wow, David and I, even though we're 50th percentile, we drew a 90th percentile kid, or this kid is, not doing very well. It's at the 10th percentile. There's not a lot of, unless someone has a serious developmental problem, there's not a lot of evidence at 6 months in the normal range of distribution. By 2 years old, you can see a kid is picked up language or is physically active and engaging or whatever. So we indeed see no effect of the kid's polygenic score at 6 months, the polygenic score for education. We've repeated this for the polygenic score for cognitive ability. Education's, the results are the same. But when we look at 36 months, so even at 3 years old, we see that the kid with the higher draw is evoking more parental attention. This is an index of all those kind of things, like how often do you speak to the kid, read books to the kid. I can't remember all the items in the index, but it's the kind of things that the Annie Casey Foundation or developmental scientists would say are good for child development 0 to 3. So we see that conditional on the parents, the draw of the kid actually affects the behavior of the parents. So to answer the sociological question, we run it again. This is an interaction with this particular one is education, but we can do with income. It doesn't matter. This is not significant, but the point, you could just see the pattern again, and we'd love to have a bigger sample size. If I remember this is not significant, it might be. Actually, it might be now that I'm looking at it. For people who are college educated, they parent. So the answer between Becker and Tomes versus what I had thought, and other sociologists, is kind of a split decision, because high SES parents basically parent the same way, at least at 36 months, no matter what their draw is. They're going to, the kid could be zoning out. They're going to read the books. They're going to read Dr. Seuss to the kid till it, even if it's not paying attention at all. Lower educated parents actually respond in the way that Becker and Tomes thought, that when there's a kid with a higher score, they invest more time and effort into that kid. So here we have a way to use genetics to answer a question that has nothing to do with genetics, is the point. So one last example, and I'm assuming this is not true anymore because of what Dan said before. This is a single sample, but this is the effect of the education polygenic score on self and the siblings of the ego's polygenic score on self educational attainment. And you see that the siblings' polygenic score has an effect on my educational attainment. When we look, again, a way to look at social dynamics within the family that are not, again, not a question of genetics, but asking about gender. When we look at sisters, it turns out that they're the ones that are driving the effect. Sisters have an effect on each other's attainment, and brothers couldn't give a crap about each other, and they have no effect. Again, this is not replicated from what Dan said. I'm going to ask him later at lunch. It sounds like that the sibling effects have been shown not to be that big anymore.
You want to say something about that, Alex?
Yeah, well, we had pretty good power in the UKB to do this using the new imputation method, and they came out basically null.
So what I should caveat this by saying this is the Minnesota twin family study. So this is dizygotic twins. So it may be that, and we might expect that for twins that actually grow up literally exactly together, there might be more of an effect than for singleton siblings. But this is probably a false positive. But
is this controlling for parents?
Yes.
Okay, that's interesting. We should talk about this.
Yeah.
If twins are different from normal sibs in this respect, then that could explain a lot of what's going on with heritability estimates, essentially.
What's that theory for the gender difference?
I mean, I went to theories of, what's her name? Help me, Paige, at Stanford, the psychologist, Carol Dweck, who talks about how girls are more, socially and empathetically engaged with each other. They have more helping behavior. I don't, but this is gender differences in development are not my expertise. I've never replicated it. This has been a slide that's been in here forever, and I've never actually done anything with it. So I just think it's really interesting. But again, an example of the kind of thing you can look at using genetic data that has nothing to do with genetics. Okay, I don't know how much time I have, but I'm going to give one or two more examples.
Okay. They don't have a schedule in front of them. I think you have more. I think you have like 20 minutes.
20 minutes? Okay. So we would think that something like race is like not something we can answer at, can integrate genetics into it all, given population stratification we heard about and so forth, and the fact that, as I'm sure you'll hear about later, there's issues about porting polygenic scores and so forth, and even just single effect sizes across ancestral populations. But I'm going to show an example of how we might use genetics to help, again, show a social effect. So in the literature, there's been a long-standing correlation between darker skin tone among African Americans, but also among Brazilians and South Africans and all over the world, among people of African descent, darker skin tone and stress and the sort of biomarkers of stress, meaning in this case hypertension. So African Americans with darker skin are more likely to have a diagnosis of high blood pressure and to have higher blood pressure as measured by a sphygmometer, or however you pronounce that. However, we know, again, because we're good social scientists, that could be any number of factors. For example, in the United States, we know that African Americans in the southern states tend to have darker skin than African Americans in the west or the north. And we know that the southern states, for all races, have many other risk factors. Like it's called the stroke belt, not because of diet and other behavioral aspects that that raise the risk of cardiovascular disease for everybody, whites, blacks, Latinos, everybody. So just one example, we could control for region, but of course, as you can think of, there could be any number of other factors that might be driving that correlation. So we want to get closer to the idea that it's a causal relationship that works through differential treatment of people by their skin tone. So the first thing we do is we use the add health siblings, and we show that if you're a full sibling, then it is a random draw whether or not you get a lighter or darker skin tone from your parents, a genetic draw. And we show that that lighter skin tone lowers the likelihood. This is an 8-point scale. It's a binary outcome, so I don't, forget about what the effect size is exactly. But basically, it significantly lowers the likelihood of being hypertensive. We also look at actual blood pressure, same effect. However, if you're good geneticists, you know about pleiotropy. So pleiotropy is when the same genes have multiple effects. And because there's so few genes in the human genome, we know that all genes are working very hard, multiple jobs, in different tissues. They're doing different things at different times of development. They're going on and off. So it could be theoretically possible that the same genes that control melanin levels in the skin have some independent effect on, say, the elasticity of your arteries or the strength of your heart muscle or whatever. We don't know. And just because you could do a search in the literature and you might not find that doesn't mean it hasn't been discovered yet. So there could be pleiotropy. So it still could be the fact that, no, actually, just because I use sibling fixed effects doesn't mean that I've discovered that there's a social pathway that's discrimination based on skin tone that causes stress levels and that causes hypertension. So what we did was we looked at, it's not a polygenic score, this was done before the recent polygenic score for skin tone, but we looked at the top SNPs for skin tone, and we looked in non-African American population, and we found that none of them had an effect. This one is marginally significant, but in the wrong direction, meaning that darker skin has a salutary effect on your blood pressure. Anyway, the point is, we used a placebo population where there's still variation in the individual SNPs, but the social mechanism that we're hypothesizing shouldn't be there. And we find, and so if there's an effect there, it suggests, well, maybe it's pleiotropic, but we don't find an effect. So we use the genetics to shoot down the sort of alternative explanation of genetic pleiotropy. Okay, I'll do one more example. Maybe some of you are familiar with the immigrant health paradox, that immigrants tend to be of better health, even though conditional on their socioeconomic status, having lower socioeconomic status in their countries of origin, and once they get to the US or the UK or whatever the receiving country is, they tend to display better health than their native-born counterparts. Certainly here, and some have argued, than the countries of origin. But of course, there's a lot of selection. There's probably not a dynamic where there's greater selection gradients in the social world than immigration. Healthier people, more motivated people are going to be the ones to immigrate. There's also what's called salmon bias, that the less healthy ones are more likely to return to their extended families and so forth in their countries of origin, and then therefore we don't measure them, their health over the long term. But it could be that there's, as many sociologists have argued, there's actually a protective effect of the culture from the country of origin. And so, for example, most of the literature has been on Mexican Americans. So the Mexican diet is way superior to, in terms of the health effects, than the Mexican American diet, or the American diet, which gets adopted by immigrant over time, so that the more recent immigrants have better health than second generation and so forth, because of McDonald's and everything else, Coke, big gulps and all the things we do here, cigarettes, et cetera, so that there actually is a protective effect from early life exposure to a different country's health regimes. So how do we separate those out? Well, again, people have looked at things like BMI before leaving, BMI after leaving, and so forth. But again, those can be affected by both social contexts. It's hard to know what's an effect and what's a cause. And with the genetics, we know that it's fixed at conception. So the simple thing is asking, do immigrants look different on their polygenic score than native-borns? And this is a joint paper, again with somebody who did this workshop, Zoya Guberski. I'm probably pronouncing her last name wrong. She's an assistant professor at SUNY Albany in sociology. So we look at non-Hispanic whites, Hispanic whites, and non-Hispanic blacks in the HRS. These are small samples. We're doing it in the UK right now. And you'll see for non-Hispanic non-Hispanic whites, they have lower genetic propensities for overweight status, lower genetic propensities for lifetime smoking and how much they're smoking. No effect, no difference on height. Among Hispanic whites, there's no significant differences, and among non-Hispanic blacks, there's negative selection. Of course, this is the slide that you should pay the least attention to because this is using the polygenic scores that were trained on Europeans. I think we got criticism that we were focusing on white population only, so we included it. What we find is that if you look at the second model, which has demographic covariates, compared to the green model, that when you control for the polygenic score, the disadvantage of height, for example, so actually I should have said that height is the one area where immigrants have a disadvantage, not an advantage. I'm sorry, this is doing its own thing. Is significantly reduced. That's a significant difference. When you look at non-Hispanic blacks, again, same thing. Okay. This is the effect on BMI. You see that the immigrant health advantage decreases when you control for the polygenic score. For non-Hispanic whites, it's kind of nothing for Hispanic whites, and it gets slightly worse for non-Hispanic blacks. For smoking, the immigrant health disadvantage in smoking increases for non-Hispanic whites when we control for their better polygenic scores. It's kind of flat for non-Hispanic blacks, and there's no effect on Hispanic whites. So many limitations I mentioned. This is a very small sample size for the kind of thing we're trying to do. It's also the HRS, so it's super old immigrants. It doesn't reflect what we're seeing today, what you do with the younger study. But I can stop there. Dan, is that good? Keep going? Okay. I'll use one. Is that a question? Yeah.
Yeah, I was wondering in terms of selection bias, so
Can you? I'm sorry. I'm hearing impaired. So if you could, yeah.
So in terms of selection bias, especially for immigrants,
Mm-hmm.
so who end up growing up and immigrating at some point are the ones I assume in terms of health and other outcomes are better off than who may not make it into the sample.
Exactly. So one of the
results that
So one of the things we want to do is look also at vis-a-vis country of origin. How do immigrants, people who emigrate, compare on their genetic traits? yeah, there's a lot more work. Was just kind of like a very initial foray into this literature, but just by way of example, how you could start to address some of these questions about selection using the genetics. The last thing I'll show is an example. I think that, at least initially, before I realized sort of how you use genetic tools to answer social environmental questions, I think a lot of us went into this field because we're interested in the idea of heterogeneous treatment effects. That, you know, we observe in the data that from a very poor crime-ridden neighborhood, you know, some kids are resilient and some kids wither in the face of that kind of environmental disadvantage. If you do a randomized controlled trial and you implement a new reading or math program in an elementary school, and you get like, wow, there's a, you know, in the two groups, it's randomized by classroom, and like the kids in the treatment classroom at the end of the year, you know, have an 8% better math score, and it's significant. You're like, that's great. I did a randomized trial, and but when you look at the data more closely, you'll see that that's driven not by an average of an 8%. It's driven, I mean, it is an average of 8% increase, but it's really driven by a few kids who did much better in that treatment regime of being taught math in a different way. And if we could identify those kids, we would know the way to teach them math, and we would teach it differently to other kids. I think you've used the example of dyslexia, like if there's a polygenic score for dyslexia, and we can actually identify kids at risk before those kind of issues emerge, we would do much better in teaching dyslexic kids to read, and they would fall much less behind other kids. And that's the kind of promise of genetics, some idea of personalized education or personalized social policy or personalized, because there are heterogeneous responses to treatments. And the intuition is that the heterogeneity in that response is largely driven by genetics. Of course, it could be driven by other environmental factors in a kid's life or in an adult's life, but the intuition of many of us who went into this was that we have the environmental treatments, but we know people respond differently, and probably that black box of how people responded differently is largely driven by genetics. And like the metaphor is the Pink Floyd prism, that you shine the white light of a treatment, and you get this rainbow of heterogeneous treatment effects. So how do you do that? Well, it's really hard because you need the environment to be randomized, and you need the genetics to be randomized, right? So I'll give you one example. I've used both in genetic and non-genetic studies that you remember from, if you're an economist, you probably read Josh Anger's famous paper that some people say started the causal revolution in economics, that uses the Vietnam era draft lottery in the United States as a natural experiment. This was a politician, congressman from upstate New York, who thought that it would be good publicity for his reelection to pick the balls to send his constituents' children, because 18-year-old males could not vote at that time, but their parents could vote, to send their sons off to fight and possibly die in the jungles of Southeast Asia. It was a good reelection photo op. But anyway, he picked the balls out with the birth dates, and that determined the likelihood of being drafted into the Army during the Vietnam War. And to top it all off, this 1972 lottery, those people were never called, so it's a nice placebo that Angrist and others have used. Just to show quickly, well, there's a heterogeneous response to being drafted. We know that, for example, the Army is one of the most tobacco-philic or whatever tobaccogenic environments you can be exposed to. That is, they literally, in combat, give cigarettes away for free to keep people alert and also calm. There's huge peer effects within your units of smoking. If you weren't a smoker before you went to Vietnam, it was highly likely that you would have smoked cigarettes there. But what the long-term lifetime consequences of that are open, and we find that there is an interaction effect between the smoking polygenic score and exposure to, meaning being drafted into the Army, so someone with a 2 standard deviation or plus 1.5 standard deviations on the polygenic score for smoking, the treatment of being drafted results on average with a 1 pack a day smoking habit that you would not have had otherwise. So that's a huge public health effect in terms of likelihood of cancer and so forth, and other emphysema and other health-related effects. Again, this was joint work with Lauren Schmitz, who I think came to this, she both as a student and an instructor, who's now an assistant professor at University of Wisconsin. So we also can look at birth cohorts, treating birth cohort, forget about mortality selection bias for a moment, and we look at across birth cohorts, the polygenic influence, in other words, the effect of your polygenic score on your behavior seems to be increasing. As we get more and more information about the ills of smoking, it's the people who are genetically more pre-programmed to smoke who are more likely to do it. Back in, if you're born in 1920, it was such a social norm to smoke, and we knew very little about the negative consequences. If you remember those ads where it said, like, it clears out your lungs, doctors say, and things like that. Helps digestion, which might be true. I don't know. There was very little correlation between your polygenic score for smoking and your smoking behavior, but you're born and you grew up, I should say, around. You're born shortly before the 1964 Surgeon General's report. The people who smoke become more genetically selected to smoke, and there's been a debate or argument about whether or not that explains, for example, why there's a declining elasticity of cigarette taxes. In other words, when cigarette taxes were increased 30 or 40 years ago, they resulted in smoking declines. But today, when they increase cigarette taxes, it does not appear. People are very inelastic in their smoking behavior, and that might be because, look, if I'm an addict, I'm going to just pay whatever it is. So it's good news for raising money through sin taxes if you're a governor or whatever. Bad news if you're trying to use that as a public health lever to further reduce smoking. But again, that's a controversial claim. Some people say there's no evidence for that, and some people say there is. So that's it. I hope that gives you a taste of how you can use genetic data, again, to answer questions that have nothing to do about how genetically determined or not a particular outcome is, or anything having to do with genetics per se, but really having to do with classical social science questions.